So now let's compare replication and transcription from a table point of view and look at what's going on in prokaryotes versus eukaryotes. So at least in transcription for prokaryotes, you see that there's a single RNA polymerase that's required. It's an alpha-2 beta prime tetramer of proteins that come together and form a complex that will transcribe prokaryotic DNA into messenger RNA. For eukaryotes, we actually have 3 separate RNA polymerases that are going to accomplish this task. There's RNA polymerase 1. You can remember that RNA polymerase 1 is going to transcribe the most abundant RNA species, which is ribosomal RNA. And don't forget that this happens within the confines of the nucleolus. That's where the ribosomal subunits are going to be put together and exported out into the nucleus. There's one key exception to this, and you need to know this. This is the 5S ribosomal subunit. For some reason, the USMLE likes the fact that the 5S ribosomal subunit is not made by RNA polymerase 1. It's the only one that's going to be made by actually RNA polymerase 3. So keep that fact in mind. Now, some other facts is that prokaryotic transcription requires a sigma factor in some cases to initiate at a promoter site. The sigma factor will come along, bind to the promoter site, and that will then recruit the RNA polymerase to bind and to start transcription initiation. Notice in eukaryotes, there's no sigma factor, but transcription factors such as TF2D can bind before the RNA polymerase is able to bind. We'll take a look at this a little bit later. Finally, for termination, the Rho protein is a protein that aids the prokaryotes in termination of transcription, whereas in eukaryotes, there's no Rho protein needed. So if you see a case where there's a Rho protein involved in termination of transcription, you can bet that they're talking about prokaryotes and not eukaryotes. Probably the most important aspect of this figure is the following. It's down here at the bottom, and you see that what inhibits prokaryotic versus eukaryotic transcription. In prokaryotes, there's a drug called rifampin, and rifampin will specifically block the alpha-2 beta prime subunits of prokaryotic RNA polymerase. Remember, it's this tetrameric protein that comes together to make a ribosomal RNA, messenger RNA, and the transfer RNA in the prokaryotes. So blockade with rifampin is going to block all of RNA synthesis in prokaryotes. What would you use rifampin to treat? You should be well versed in this. The key disease that comes to mind is, of course, tuberculosis, but also you can use it to prophylax for meningitis. So a typical case is to give you a patient with tuberculosis. They're having night sweats, they're perhaps having hemoptysis, coughing up blood, a lung x-ray is done, and there's granulomatous nodules seen on the chest x-ray. The patient is treated with a particular type of drug. What's the mechanism of action behind this drug? Well, for rifampin, it's going to be blockade of the alpha-2 beta prime subunit. Now, in the case of meningitis, remember that rifampin is not used to treat primary meningitis. It's used to prophylax in close household contexts of people that have acquired bacterial meningitis. And one of the hallmarks of rifampin, one of the side effects of rifampin is what? Remember, it's the red-orange colored urine. As a matter of fact, all body secretions, tears and saliva can even have an orange colored tint to them after rifampin treatment. While we're on the subject, can you remember the 3 other drugs that are used to treat tuberculosis? Not necessarily, they're not going to block prokaryotic transcription, but what are those other drugs? Well, there's 4, one of which is rifampin. The other 3 are pyrazinamide, ethambutol, and isoniazid. You'll hear about an isoniazid later on in this review. As far as eukaryotes go, there's only one real inhibitor that we have to concern ourselves with, and that's alpha-amanitin. Alpha-amanitin is a toxin that's made by a certain type of mushroom that grows in the wild. It's called the death cap, or the Amanita phalloides mushroom. Now, mushroom picking may be a hobby of some, but if you're going to pick mushrooms and eat them, you better know what you're doing. Because if you pick one of these mushrooms and you eat them, the toxin, the alpha-amanitin toxin, will completely ruin your liver. You'll go into liver failure and die if you don't get a liver transplant. Now, why does this happen? It's because the toxin from this mushroom is going to specifically block the eukaryotic RNA polymerase. In fact, it blocks RNA polymerase 2, so you should know that it's going to block messenger RNA synthesis specifically. Not ribosomal RNA, not transfer RNA, but messenger RNA synthesis. As far as actinomycin D goes, this is more of a laboratory experimental type drug. It isn't something you'd be asked about on the board exam.